Yes, guys, am I? Yes, I think it's fine, right? Yes. Okay. Cool. Perfect. So we'll just wait for everyone to come along. Just wait for like 10 seconds or something like that. Okay, so that everyone can join the live. I think I'll just put down the comments section as well. Okay, perfect. I think slowly momentum will be coming in. Hello, Macha. Hello. Okay, so yes. Guys, did you hear me out for this particular question? Until here it was clear, right? Until here it was clear, I assume. Yeah? Thank you so much, Swami. Okay, so we'll move on to the next question then. Perfect. So I do not know until when did it got stopped, but yes. Question number 34, here it goes. We are not going to be stopped. We are unstoppable, right? Yes. So in zygote, intra-fallopian tube transfer, the embryo up to dash stage is transferred into the fallopian tube. What do you think is the answer for this question? 8 blastomeres, 32 blastomeres, 2 blastomeres, or 16 blastomeres? What do you think is the answer for this particular question? Any idea? In zygote, intrafallopian transfer. Zygote, intrafallopian means you are putting the zygote inside the fallopian tube. But here it's not just a zygote, you can put embryo also. And most of you guys are saying A is the answer. Okay, starting with Bhavana, Amalu, and Tarun, and Meenaz, and also Karthik, Geeta. Everyone says A is the answer. A is what? 8 blastomeres. So Baba, how can you say that, ma'am? It is zygote, no? But listen, ZIFT, zygote intrafallopian transfer, it means that it can be either zygote or it can be also the embryo. If it is the embryo, it has to be only up to 8 cell stage, not beyond that. If at all it is beyond that, it will lead to what? Ectopic pregnancy. That means pregnancy at areas different from the uterus, and it can lead to that of the mother as well as the child. So which is why the answer for this question is going to be 8. One more very important question to you guys. If at all it is more than 8 cell stage, where do you put the embryo? If at all it is more than 8 cell stage, where are you going to put the embryo in? Not fallopian tube, then where? Yes, check motivation. Geeta, Amalu, first leg, thank you so much, dear. And Kumar, Deena, Prakash, Sanhit, everyone out there, you're doing absolutely great. Okay, so yes, uterus, exactly. If it is more than 8 blastomeres, you are going to put it inside the uterus, because that is where pregnancy has to happen. Yeah, so the answer for this particular question is going to be 1. And if it is clear, can everyone give me a quick thumbs up? Can everyone give me a quick thumbs up if at all it is super clear to you? Yeah. Thanks, Macha. Thank you so much. I'm confused in reproduction chapter. What do? Baba, if at all you just go through the one shot of it, it will be really helpful if at all you're too confused. Yeah? There is masterstroke video. You can check it out. Once more, ma'am. Manjula says once more. I will explain once more only. Sorry. So in zygote intrafallopian transfer, Baba, you're going to put the zygote inside the fallopian tube. Now listen, it is not just zygote. It can be embryo also. Only if the embryo is in 8 cell stage. Okay, if it is in 8 cell stage, you can still put in the fallopian tube. Why? Because when you put in the fallopian tube, slowly it is going to divide, and it will roll down automatically inside the uterus, and in the uterus only pregnancy will happen, right? But if at all you are putting an embryo which is more than 8 blastomeres into the fallopian tube, it can lead to ectopic pregnancy in the fallopian tube itself, and that can be dangerous for the mother as well as the child. Okay, so yes, so that is the answer for this. I hope that is clear. Now we go ahead with question number 35. Statement number 1 says, MTP is to get rid of wanted pregnancies due to casual unprotected intercourse or failure of contraceptives used during coitus or rapes. Statement number 2 says MTPs are performed legally by qualified doctors by giving proper medical justification. What do you think is the answer for this particular question? So previous year a lot of you got confused. I mean, not you, but the children got confused seeing this question. Please make sure you check it out carefully. I don't know, Macha Prince, some technical issue, some network went down, so I think it just got off. Yeah, so Quadric says B or 2 is the answer for this question. Amalu says D is the answer. Karthik says B. Hassan is also there. Hello Hassan. Says C. GS World says B. Bhavana says B. Shankar, Bhavish and Tarun, Chetu, Melo and Milan. Okay, but some people have different answer, but yes, most of you are saying 2, that is basically B is the answer for this question. Statement number 1 is incorrect, but statement number 2 is correct. Is that so? Let's check it out. MTP means medical termination of pregnancy. Baba, it is to get rid of wanted pregnancy. No, it is to get rid of unwanted pregnancy. Why will you terminate a pregnancy if at all it is wanted, right? So this will be wrong. Rest everything is correct. MTP is to get rid of unwanted pregnancy due to casual unprotected sexual intercourse or failure of contraceptives during coitus or rapes. Rest everything is correct. Just they have made this particular thing wrong. It is not wanted, it should be unwanted. Now, statement number 2. MTPs are performed legally by qualified doctors. Yes. So in NCERT it is written, it is performed illegally by unqualified doctors. Yes, it is performed illegally by unqualified. It is performed legally by qualified doctors only, right? So this particular statement is going to be correct. So statement number 2 is correct, and statement number 1, which is there, it is going to be incorrect, which is why the answer for this question is going to be this. Is this clear to everyone? If yes, just give me a quick thumbs up. Salam walaikum. Walaikum assalam, dear. So I do not know if the response is correct, but this is what I've heard. Yeah? Correct? No. Let's go ahead with the next question. Yes, perfect. Thank you so much, guys. Question number 36. How many types of gametes will be formed by the parent with genotype capital A small a, capital B small b, capital C small c? Ma'am, this is not in our NCERT. How will we know? You will know everything. Inchara, good afternoon, dear. Okay, just keep on going. You will be able to do it. How many types of gametes will be formed by parent with genotype this? Wanted, no wanted. Yes. A ka 1. A1 now. Okay. A1. Oh, you're answering like that, Bhavish. Okay. So Bhavna says A, Abhishek says A, Karthik A, Melo says A, Bhima says D, Anjali says A. Okay, guys, check it out. Rihan says it is 3. Geeta says A, Bhavan says it is A. Okay, Ritika says it is A. Most of you guys are saying it is A. Some people are saying it is B also. How do you find the answer? It's very simple. First of all, spot the number of heterozygotes. Heterozygotes means basically the alleles combination has to be different. For example, when I say it is like this, it's homozygote condition, right? It is not heterozygote. When I say it is like this, it is heterozygotic condition. So over here, this is one heterozygote, this is another heterozygote, this is another heterozygote. 3 heterozygotes are there, capital A, capital small a, capital B, small b, capital C, small c, right? 3 heterozygotes. Now you have a formula, 2 raised to the power n. N is number of heterozygotes. N is nothing but the number of heterozygotes. So 2 raised to the number of heterozygotes is what? 3. 2 raised to the power 3 is what? 2 into 2 into 2 is equals to 8. It is equals to 8. So your answer for this question is going to be 1. Please do not forget this formula. Amalu, 2 power n, exactly. Okay, so everyone who got your answers correct, kudos to you. For everyone who got your answers wrong, I hope you have learned something new today. Yeah? Yes? Clear? Thumbs up. If it's super clear, just give me a quick thumbs up. We will move on to the next question. Okay, and I'm so sorry for the live to be ended. Didn't expect that to happen. I was teaching myself only. You know what? Ma'am, my answer is wrong. Bhima, it's okay, da. At least you learned something new, no? You know the answer now, right? So be happy. Yeah? Yes, perfect. So let's go ahead with the next question out there, and it goes like this. Question number 37. When a single gene exhibits multiple phenotypic expression, the phenomenon is called. A single gene is there. It is basically controlling lot of different phenotypic expression. What is the phenomenon that is being applied over here? Any idea? Pleiotropy, codominance, polygenic inheritance, incomplete dominance. Any idea? C. Okay, most of you guys are saying it is A, but some of you are also saying it is C. Why C? Let me see what is C. Polygenic inheritance. Okay, Pleiotropy. Listen, yes, I definitely knew that you would be confused between this and this. Listen to me very carefully. Both of them start with P, but there is a difference out there. Pleiotropy is the answer for this question. Example is phenylketonuria. You know there is an enzyme. For example, I say this particular gene was coding for the enzyme phenylalanine hydroxylase. This is an enzyme, okay? What does this phenylalanine hydroxylase do, Baba? It is the one that is responsible for conversion of phenylalanine, that's an amino acid, to another thing that is known as tyrosine. Okay, so when this enzyme is not there, why? Because some gene mutation has taken place over here. When this enzyme is not there, this conversion is not going to happen. Yeah. What happens if this conversion is not taking place? The thing that happens is that, first thing that happens is that, basically phenylalanine is going to be accumulated in the form of phenyl pyruvic acid. Ma'am, what's the problem with accumulation? If it is accumulated, it can lead to mental retardation. It can lead to what, Baba? Mental retardation. So this is one character that is being affected. The second character that is being affected because of the same gene is that, basically, if tyrosine is not being formed, tyrosine is super important for the formation of our skin pigment, skin or hair pigment and everything like that. Skin and hair pigment, which is why we will be having hypopigmentation. Your hair might be gray in color, your skin might also have a different color. That is hypopigmentation, or reduced pigmentation is going to be seen over here. So Macha, ma'am, you are saying that because one gene is affected, multiple characters are also being affected? Yes, this is an example of what? Pleiotropy. This is example of pleiotropy, and that is your exact question. Now some children had doubt between pleiotropy and what? Polygyny. Polygyny. So listen to me, polygyny is another thing. Totally different thing. Polygyny means that, for example, I'm talking about my skin color again, or probably height. You know, your skin color and my skin color is totally different. My skin color will be different from all the other people who are there in this office also. Why do you see different variation in that? Because skin color is not coded by one gene. It is the multiple effect of multiple genes out there. So basically, if at all I want to give you an understanding of this, I can say, suppose a gene is there, capital A, capital A, capital B, capital B, capital C, capital C. According to Mendel, he said that only this is going to code for one particular character. But here, the combination of this is going to code for your skin color. So if at all your genotype is like this, it is going to give the darkest skin color out there. If at all, on the other hand, you have a recessive allele out over here, it is going to be capital A, small a, capital B, capital C, capital C. It is going to be lesser color. That means your pigment will be lesser than the previous one. Okay. If at all of them are small a, small b and small c, it is going to be the lightest skin color. What do you understand from this? Multiple genes, polygene, poly means multiple. Multiple genes has effect on one character. Pleiotropy is the ulta. One gene has effect on the multiple characters. So imagine, do not, what do you call, do ratification, understand the concept and solve the question. Yeah, which is why the answer for this particular question is going to be 1. Is this thing clear to you? Every one of you who is watching this video, clear? If it's clear, just give me a thumbs up. Yeah. Madan Reddy, thank you, Macha. Okay, and everyone, Yanvi and Guru Bro and Rohit, Madan and Shankar, Prince, Bhima, Geetar, everyone who gave me the answer, thank you so much. Ma'am, principle of inheritance. So basically, I'm doing a previous year question paper here, so we will be seeing questions on the principles of inheritance and variation as well. This is one such question that we have, right? Yes, perfect. Let's go ahead with the next question. Question number 38. Another question in principles of inheritance and variation. A colorblind man marries a carrier woman. The percentage of their colorblind progeny in the next generation will be what? The percentage of their colorblind progeny in the next generation is going to be what? Any idea? D. Okay, Shankar says it is D. Quadrikit says it is D. Kavya says D. Bhavana says it is D. Sarveen Kumar says it is A. Okay, Amalu says it is D. Malo says it is D. Abhishek 2. Okay, it is B. Usman says it is D. Most of you guys are saying it is D. Passionate, Passionate and Bhima. Okay, Chandana is also there. Deena is also here. Listen to me out carefully. So, if at all we solve these questions, it goes like this. Colorblind man. A man can be colorblind only if his X chromosome is affected. right? He cannot be a carrier. Males cannot be a carrier if in sex-linked disorders, right? What about the female? Female is a carrier over here. That means only her one X chromosome is being affected. You are being asked about their colorblind progeny in the next generation, percentage of colorblind progeny, not carriers, colorblind progenies, right? So this and this goes, I get XXXC. What do you mean by that? She is going to be a carrier. She is not having the disease. She is a female, okay? She is not having the disease. She is just a carrier. This and this goes, I get XCXC. That means she is having the disease, okay? She is going to be colorblind, and this is again going to be a female, okay? This and this goes, I guess XY. It's going to be a very normal guy because none of the genes are being affected. Only if X chromosome is affected, he will have the disease. Now you have this and this. XCY. Over here, one gene is affected and only one chromosome, X chromosome, if it is affected, that is enough for the males because they do not have anyone to overshadow the effect over here. So this guy is going to be colorblind and this is a male. So in out of 4, how many of them are colorblind? This lady is colorblind and this guy is also colorblind. Out of 4, 2 of them are colorblind. So basically 1:2, that is half of it, 50% of them are going to be colorblind. Your answer is going to be 4, that is D. So everyone who said D is the answer for this question. Everyone who had a different answer, I hope it's clear as well. Akhil, BMS and Lahari and Aniket and Tarun, everyone. Guys, if at all you got the answer wrong, please make sure you check it up. Okay, ma'am, when will you do complete NEET? Dear, so teachers are doing NEET live classes out there. Please make sure you check out their video. So since since KCET is also upcoming, which is why I'm more inclined to that. But yes, you can check out the other teachers' videos out there. They are really helping you well, yeah? Yes. I hope this is clear to everyone. If yes, can you quickly give me a thumbs up? Ma'am, repeat once again. I can repeat once again. So listen, over here, you have been asked about colorblind man. Colorblind man means his one X chromosome is going to be affected, right? Okay. What about lady carrier female? Carrier female means only one X chromosome has to be affected. The other X chromosome will overshadow the effect. That is why the person will not have the disease. Now, when you do the crossing, you will be able to see that when you get the first one, it's going to be a carrier female only. She will not be having the disease. Why? Because she has a normal X chromosome that will overshadow the effect. She will be just a carrier. She is not having color blindness. But on the other hand, when this chromosome and this chromosome comes together, you get a color blind female. Both these chromosomes are affected. When this chromosome Y, and from here you get a normal X chromosome, it's a normal guy that is being formed. From here you got Y, and from here you got an affected chromosome, that is XC. Again, you are going to get a male. Over here, no one is there to overshadow the effect of this, which is why this guy who has one affected allele will be having the disease out there. So out of 4 of them, how many has the color blindness? That is the question. Percentage of colorblind progeny. Out of 4, 2 of them have color blindness. One is a carrier, one is a normal thing. Rest, both of them are colorblind. So 2 out of 4, if at all I cut it off, it is half. 2 out of 4 means half of the population, right? So it is 50% of them will be colorblind. Cool? Works? Ma'am, please explain slowly. Okay, done. I'll explain slowly. So sometimes when I go slow, which I think I was in the previous live, okay, so their children said fast, so that's why. Yes. Cool. Now I'll go slow. Fine. Question number 39. Identify which one of the given pair of options is correct with respect to Down syndrome and Turner's syndrome. Down syndrome and Turner's syndrome. Come on. Try it out. A, B, C, D options are given to you. Short-statured individual for Down syndrome. In Turner syndrome, gynecomastia in man. What do you think is the answer for this? BMR says the answer is A. Okay, Kodrik it said it's A. And Pratna says it is A. Bhavana says it is A. And okay, Rohit Kumar. Okay. A. Most of you guys are saying it is A. So I have to check out, are you guys giving me the answer from here or there? Don't do the mistake. Again, I'm telling you. When you do the mistake over here, you will do the mistake in the OMR sheet also. Check your answers from here. Give me the answer like 1, 2, 3, 4. Give me the answer like 1, 2, 3, 4. It will be really good. Okay. In Down syndrome symptoms, short-statured individual, definitely there. Turner syndrome, gynecomastia in man, wrong. Gynecomastia in man is found in Klinefelter syndrome, not Turner syndrome. So this is wrong. Now, Down syndrome, round head and partially open mouth. Round head and partially open mouth in Down syndrome, correct. Turner syndrome, overall masculine development. Is that being seen in Turner syndrome? No, Baba, that is again seen in the case of Klinefelter syndrome. They have overall muscular development, but they also have breast, that is gynecomastia, right? Now, broad palm physical and mental development is retarded, correct. Sterile females with rudimentary ovaries, correct. Turner syndrome, they just have one X chromosome, another X chromosome is not there, which is why they will have rudimentary ovaries. Their ovaries are not well developed, which is why they will not produce ovum also. That is why we call them as sterile females. This is a character that is correct for both of them. C1 is the correct for both of them. If C is correct, your option is going to be 1. Your option is going to be 1. Additional copy of an X chromosome in Down syndrome? No, it is additional copy of 21st chromosome. Absence of X chromosome is correct, but this is wrong, right? So which is why this option is wrong. Your only answer for this question is going to be 1. Your only answer for this question is going to be 1, that is C. Guys, can you give me the answer like 1, 2, 3, 4, because I think that is how it is, the question is being asked, right? So can you try to do that? It will be really, really good. If this question is super clear to all of you, can you give me a thumbs up? Thank you, Abuva. Everyone else, Anand, thank you, dear. Okay. Guys, from now on, give me the answer like 1, 2, 3, 4, okay? Yes, perfect. Okay, question number 40, that is RNA polymerase 2 is responsible for the transcription of. RNA polymerase 2, Baba, is responsible for the transcription of what? hnRNA, snRNA, tRNA or rRNA. What do you think is the answer for this particular question? Okay, Rihan says 1 is the answer. Quadric says 1. Amalu says A. Okay. Rihan says 1. Bhima says it is 3. Amalu says 1. Sanjay says it is 2. Okay. Aniket says 1. Kalpana says 1. Salena says A. Okay. Most of you guys are saying it is 1, but some people are confused between 3 as well, which is why I have to check it out. RNA polymerase 2 is responsible for hnRNA transcription. Listen, if at all you do not remember it, listen to me out carefully. There are 3 types of RNA pol. Okay, RNA pol 1, 2 and then finally 3. RNA pols 1's primary, okay, the first preference is given to, the first preference is given to formation of rRNA itself, because it starts with RNA, R, no? Same, so preference wise also rRNA is given the preference, and over here different types of RNA is being formed. 28S, okay, 18S, 5.8S, all of them, all the different types of rRNA are being transcribed with the help of what? RNA pol 1. So how do I remember 28S, 18S, 5.8S? Because everything ends with 888. If at all you look at this, everything, Baba, is going to end with 888, right? Yes. Guys, Solinity, please try to switch off your mobile and restart it once. Okay. Next one, RNA pol 2 is the one which is responsible for, second priority is given to hnRNA, formation of hnRNA. You know, by transcription, we have DNA transcribes to hnRNA, and then later it forms the mRNA. So the precursor of mRNA is hnRNA, that is being formed with the help of RNA polymerase 2. Now, RNA polymerase 3 as in T, 3 as in T, so T with 3 and T with tRNA. So tRNA is going to be transcribed with RNA polymerase 3. Apart from it, there is small nuclear RNA that is also transcribed with the help of this. Apart from it, there is one type of rRNA which is also transcribed. Okay, one type of rRNA which is known as 5S rRNA. So here everything was 0.8888, right? So over here it was not 8, it is just 5S rRNA that is also transcribed with the help of this. Please remember this, it's super important. Okay, it is going to be super important. Is it clear? Will you be able to remember this? Try to remember it, okay? It's going to be looking like a bit tough out there, but volume problem is there, guys? I don't think so. Is there, guys? I don't think so. No. So volume problem is not there. Please check it out. Okay? Yes. Thank you, Macha. And if at all you want to take a screenshot of this, please take. It's important for you. Do not forget it. Have the logic remembered. Okay. Question number 41. Thank you, guys. Okay, question number 41 goes like this. Which of the following enzymes increases the permeability of the bacterial cell to lactose? Which of the following enzyme increases the permeability of bacterial cell to lactose? Transacetylase, amylase, beta-caroteridase, permease. What do you think is the answer? You have to think. 4. Rihan says 4 is the answer. Okay. What is Quadrikit says 4. Bhavana says 4. Aniket says 4. Karthik says 4. Harish also says 4. Okay. Kumar 4. Safwan says Anand. Janavi. Hello dear. Amalu says 4. Quadrikits permeates. Okay. So most of you guys are saying 4. Some of you are also saying it is 3. Okay. Some confusion is there. Is it? So always remember you have Zya. Okay, you remember Zyada we say in Hindi, right? So Zya is there. 3 type of enzymes are there. This gene is going to code for beta galactosidase. Okay, Y codes for permease and A codes for transacetylase. Transacetylase. So what is the role of beta galactosidase? It basically says that, hey, lactose, macha, you come over here. I will break you down into what? Glucose and galactose. That is how the E. coli gets the energy. If at all it gets glucose, that is how it gets the energy. So what's the best way to get the glucose? Break down the lactose. Now, Baba, this is E. coli. E. coli has to make sure that the lactose which is there, it is getting inside also. Then only this breakdown can happen. For that, you require permease. Permease increases the permeability of the membrane, and that is why you will be getting what? You are going to get the things getting inside. Now, what is left with? We are left with transacetylase. They are the ones which are going to deal with any toxic things which are going to be formed in the entire process. Okay, so the byproducts which are there, they are going to be probably dealing with all those things are there. Cool. So the answer for this particular question is going to be permease, because the question is which increases the permeability of the bacterial cell. So answer is permease. Clear, Macha? I'm suggested a study plan for exam to chemistry. Oh, yo, I will be coming up with a study plan. Zay, please do not spam over here, dear. I have to think about it. You know, creating a study plan is not at all easy. Executing is also very difficult, obviously. But yes. Guys, clear, right? Okay, shall we go ahead with the next question? Yes, perfect. Thank you so much. Next question that is there is 42nd question. Which of the following statements are correct with reference to prokaryotic genome? Prokaryotic genome, okay? So, monocystronic structural genes, introns absent in the structural gene, transcription and translation are coupled processes. Primary transcript undergoes splicing, only one RNA polymerase is present. So, you have to tell me which one is correct, okay? Regard through prokaryotic genome out there. Yes? Can we practice any chapter practically? Eh? What do you mean by that? Okay. Okay, so most of you, Veera Bhadra is saying it is 4, and Quadrikit says it is 4, Karthik says it is 2, Sanjay says it is 4, Geeta 4, Quadrikits, Apurva says it is D as in 4, okay, Vardharaj says it is 2, Siddhalinga says it is 4. Most of you has confusion? Is it there any confusion? Yes, now I see most of you are saying it is 4. 4 is what? Only B, C and E are correct. Let me check it out. Are you ready? Let's be ready. Monocystronic structural genes. What are we talking about? We're talking about prokaryotes. And we have to find what is correct. They are not monocystronic. Monocystronic means, for example, I say that this is a cystron, this will have only one gene, and this gene is going to code for only one protein. This is what you mean by monocystron. But in the case of prokaryotes, as you saw in E. coli, they are not monocystronic, they are polycystronic. P for prokaryotes, P for polycystronic. What happens there? You saw that. Z, Y, A gene. Everything was together only. 3 of them coding for 3 different things, right? So they are polycystronic, Baba. They are not monocystronic. They are going to be polycystronic. Wrong. So introns absent in the structural genes. Exactly. Listen, in the case of prokaryotes, you do not have introns. What are introns, by the way? So the DNA have some coding regions, okay, and have some non-coding regions also. All of them are transcribed as such in hnRNA. Okay, so they will also have some non-coding regions. They will also have some coding regions. These non-coding regions which are there, they are known as introns. And the coding ones which are there, they are known as exons. Further, when you form an mRNA, which is going to undergo translation later on, which is going to form proteins later on, you make sure that you remove the introns and you just have the exons over here. This process is known as splicing. It is known as what? Splicing. But in the case of prokaryotes, they do not have introns at all. They do not have introns, so do they need splicing? No. So introns absent in structural genes, correct. B is correct. Now transcription and translation are coupled processes. Is it? Yes. What do you mean by coupled processes? Are they coupled? No, Baba. That means that they are going to happen together. In the case of prokaryotes, everything happens in the cytoplasm itself. There is no nuclear membrane and stuff like that. So we say transcription and translation can happen together. In the case of eukaryotes, we have nucleus where only transcription happens. We have cytoplasm where only translation happens, right? But here it's not like that. Everything can happen together. So this is also correct. Primary transcripts undergo splicing. Splicing is not needed. Why? Because introns are not there. So this is wrong. Now only one RNA polymerase is present. Yes. So ma'am, we studied about RNA pol 1, 2, 3. That is not for prokaryotes. No, that is only for eukaryotes. RNA polymerase is only one in the case of prokaryotes, which is why I'll say B, C and E are the only options correct, which is where fourth is the answer. Cool. Got it. Shall we go ahead with the next question then? Shall we go ahead with the next question then? In which organism contain monocystrons, Bab? Monocystrons are found, monocystronic thing is found in us, eukaryotes. For example, animals, plants, fungi, we are all eukaryotes. We have monocystronic gene. Okay. In the case of bacteria, which is a prokaryote, it does not have monocystronic. P for polycystronic. Got it? I hope that works. Yes, perfect. Thank you. Next question. When a change in the gene frequency of a population occurs by chance, it is called. When a change in the gene frequency of a population occurs by chance, what do you call this? By chance, okay. Genetic recombination, genetic drift, founder's effect, and gene migration. Rihan says 2 is the answer for this. Okay. Anand says it is 2. Quadrikit says it is 2. Aniket says 2. Bhavana says 2. Karthik says B. That is 2. Manoj says A. Amalu, Baba. Okay, thinking. Rihan. Varadraj says 2. Bhima says 2. Ramya says 2. Janvi also says 2. Okay. Sarveen, Madan Rao, and a lot of other. Madan Reddy is there. Amalu is there. Okay. Nauman, hello. Okay. So Noha Kazi, everyone is back. So the answer for this particular question is going to be 2, genetic drift. Why not genetic recombination? That is also occurring by chance basically, but it happens in sexually reproducing organisms out there and stuff like that. Founder's effect is an effect of genetic drift. Because this happens, it can lead to founder's effect. Gene migration is not, what do you say, it is not occurring by chance. For example, I feel that today I'm in India, tomorrow I want to go to America. It's not by chance. I am thinking about it and then only I'm going to do that. Basically, when I go there, the allelic frequency changes. Here also, when I move out of this, the allelic frequency changes, right? So genetic drift is the only thing that occurs by chance. For example, over here, I have white beetles and I have green beetles. Okay, so white ones were more in population. They were saying that green ones say, Macha, you get out over here, you're not needed over here, you're found in very less amount. And one big day, what happened? One elephant came and stamped on this region where all the white ones died. So the allelic frequency is changing, the frequency of the alleles are changing by chance. Yes, that is known as genetic drift. So the answer for this is 2. Always remember, random change, genetic drift. Okay, clear na Macha? Okay, if it's clear, can you give me thumbs up? Founder's effect as in, Baba, so these organisms which are there, okay, these organisms which are left behind, they are going to be the population out here. They're going to be the founders of the new area out there. They are going to divide themselves and increase. Okay, so that is what you mean by founder's effect. They're the only ones which are left back. Thank you, Madan Reddy. Thank you, Hasina. Hello, ma'am. How are you? I'm really good. Thank you for telling me, asking me that. Yeah? Yes. Okay. Perfect. Let's go ahead with the next question out there. Darwin's finches represent one of the best examples of, now this is the easiest question in your KCET question paper. Darwin's finches represents one of the best examples of what? Chemical evolution, genetic equilibrium, seasonal migration, or adaptive radiation. What do you think is the answer for this? Sina, thank you so much, dear. Okay, 4. Rehan says 4 is the answer for this. Okay. Quadricid says 4. Sanjay says 4. Anand says 4. Sridhar says it is 2. Deepa says it is D. That is 4. Quadricates. Okay. Aniket and Ramya, Bhavana. Okay. Norman, Bhima, Kalpana, Manoj, Abhishek. Sorry. Janvi. Okay. So most of you are saying 4, and 4 is what? Adaptive radiation, exactly. So Darwin's finches, which were right over here, Baba, they in the Galapagos Island, when suppose I was on Darwin, okay, I am Darwin. Darwin moved this side and found, oh my God, same finches but different beak. Oh my God, same finches but different beak. Again, he moved this side. Oh my God, same finches with different beaks. So what is happening? Basically, they had a common ancestor over here. Probably they had some changes between them only. And when some of them moved this side, they were able to adapt there better. They were able to eat there better with a different type of beak and everything that they had. This is known as adaptive radiation. They radiated and they got adapted there. Okay, so the answer for this question is going to be 4. Cool? Yes, Varadraj. I will be trying to do the revision for PU2 and PU1, both of them. Yeah, mostly PU2, but yeah. 45th question. Are we ready to do it? Thank you so much, Aniket. Wow, ma'am, your teaching is beautiful like you. Oh my God. Okay, thank you so much. So choose the correct statement from the following. Charles Darwin traveled around the world in a ship called HMS Beagle. There has been gradual evolution of life forms. C. According to Darwin, fitness refers to physical fitness only. D. Fossils are remains of hard parts of the life forms found in the rocks. E. Hugo de Vries, a naturalist, worked in Malay archipelago. What do you think is the answer for this particular question? Any idea? Ma'am, next live class is going to be happening to, if at all you're asking for me, it's happening tomorrow, okay? Yes. You're the nucleus of the class, controlling everything perfectly. Oh my God. Okay, thank you so much. Okay, guys. So I see 1, 1, 1. Quadric said 1. Sridhar says is 3, and Bhavana says 3. Alima says 1. Okay, Melo says 3. Karthik says 1. Spurti says hello. Spurti says 1. Okay, Noha Kazi and Short Songs and Apurva, Anand, Muhammad Atif. Hello, dear, and Shankar, Kumar, and everyone out there. Most of you guys are saying the answer for this particular question is going to be 1. 1 is what A, B, and D are correct. Let's check it out. You have to find what is correct from the statement, right? Charles Darwin traveled around the world in a ship called HMS Beagle. Correct statement, so A is correct. There has been gradual evolution of life forms. Yes. Very slow evolution, right? Very, very slow evolution has been seen. So that is correct. According to Darwin, fitness refers to physical fitness only. No. According to Darwin, he said fitness refers to reproductive fitness. Okay, reproductive fitness. That means if at all your organisms are there and they're reproductively fit, basically they can give rise to more organisms and the population will continue. So this is going to be wrong. Now, fossils are remains of hard parts of the life forms found in the rocks. Is that so? Fossils are remains of hard parts of the life forms? Yes. Hugo de Vries, a naturalist, worked in Malay archipelago. No, he did not. He did not work in Malay archipelago. This is wrong. Until this was correct, but this is wrong. So the only answer for this particular question is going to be A, B, and D, and as you said, it is going to be 1. And with that, I have one question to ask you. Tell me, who was the one who worked in Malay archipelago? Can you tell me who was the one who worked in Malay archipelago? Any idea? Any idea out there? Also, my next class, I think I'll just check, okay? Tomorrow, the time is going to be Alfred Wallace. Yes, you guys are correct. Alfred Wallace was the one who worked in Malay archipelago, and my next class will be 9:30 a.m. tomorrow morning. Is what I assume. Everything, if you everything, everyone who have not joined the WhatsApp channel, join it right now, because there only I will be sending the message that I am coming live at 9:30 a.m. or 5:30, either way. Okay. So please check the WhatsApp channel. Please be there in the WhatsApp channel. It is definitely going to be super helpful for you. Yeah. Cool da, macha. Works na. Let's go. Okay. Everyone, by the way, who have not hit on the like button, click on the like button. Why? Because it makes sure that it goes to everywhere. Okay, when the number of likes are increasing, it moves to the every part of the place, and more children would be able to watch it as well. Okay, it increases the visibility. So please do that. Let's make it 100 here also. Yeah, okay. WhatsApp channel link, it's there over here in the description section, but if at all you do not know, I'll just copy it and I'll put it over here. Okay. Alfred Wallace, yes guys, you're absolutely correct. It is Alfred Wallace who worked in Malay Archipelago. Yes, perfect. I've sent you that also. Okay. Perfect, let's go ahead then. Let's not waste time. I know your time is precious. Okay. Question number 46. In which of the following HIV replicates and produces its progeny viruses? Killer T lymphocytes, suppressor T lymphocytes, helper T lymphocytes, memory T lymphocytes. Where do you think HIV is going to replicate and produce the progenies? You're most welcome, Geeta. No, ma'am, it's Braino Corner. Okay, so yes, Rehan says the answer for this question is 3. Okay, Neha says it is C. Passion Art says it is C. That is 3. Okay, and Bhavana and Noha and Manoj and Anant and Rehan and Mohammed Javeed, hi dear, Komali and Komalis, and I think Ramya, Spurti, everyone says it is 3. Alima, Melos and Apurva, Nauman, okay, Geeta. So the answer for this question is going to be 3, that is helper T lymphocytes. Listen to me very carefully. You have the HIV virus. The first thing that it does is it comes and attacks the macrophages. Macrophages is actually known as the HIV factory. Why? Because this is the first place to attack. Okay, once they attack, they make so many progeny virus here only, and the progeny virus is going to attack the actual target cell. What is the actual target cell? It is going to be your helper T lymphocytes. Why helper T lymphocytes? You know the name, helper. It is going to help us in building our immunity. It is like a mediator that is there. Now the viruses, if at all they are going to target the helper T cells, our immunity will go low. Okay, our immunity will go low. Now over here, ma'am, you're saying it is macrophages where they divide, but I do not have an option like that. Yes, you do not have an option like that over here, which is why the next answer that you should consider will be helper T lymphocytes. This is the place where they're going to replicate and are going to produce their progeny virus. But if at all you're being asked this question, that which is the HIV factory of the cell, you should know the answer for this. What is the HIV factory? It is macrophages. What is the target for HIV? It is helper T lymphocytes. So please know that. Why not killer T lymphocytes? Because that is not the place where they are going to replicate and they are going to produce their progeny virus. They will be, but majorly, if at all we say, it will be this. Thank you, Sridhar. Yes. Thank you, Sridhar, for following the WhatsApp channel. It is definitely going to be helpful for you guys, yeah? Yes. Perfect. So I think we are good to go with the next question out there. Hello, Manjula. Guys, shall we go with the next question? Yes, thank you. Ma'am, chemistry also you only teach. For chemistry, you have a good teacher out there. Ekta ma'am. Okay, please check her classes. Okay, question number 47. Which of the following are the techniques for detection of cancer of internal organs? By the way, guys, I don't think tomorrow I have a class. Sorry, if at all you think it's day after tomorrow, I will be having a class for FLT, full length test, okay? Not tomorrow. Yes. So which of the following are the techniques for detection of cancer of internal organs? What is your answer? Ma'am, doubt. Yes, Prince, tell me, what is the doubt? Hasina says 3 is the answer. 3 is what? A and B. A and B. Okay. What about others? Bhavna says it is 2. And Spurti says it is 3. Quadrikit says 3. Apurva says C. Okay. Rihan says 3. Chetan says 3. Madan says it is ma'am. Yes, tell me, dear. Yes. Ma'am, we want previous year KCET question papers. It is there in their website itself. Check it out, dear. Okay. Perfect, Raghavendra, perfect. So most of you guys are saying the answer for this question is 3, but some of you are also confused with 2. Guys, check out. Listen to me carefully. You have to find the cancer of internal organs, right? Okay. So you have 3 techniques actually. One is radiography. where you simply use X-rays, you know, you get X-rays and stuff like that, right? Now, the next thing is computer tomography. Again, the rays that you're going to use is X-rays only, but you get three-dimensional image of the internal organs, 3D image of the internal organs, okay? But the rays that you're using are X-ray only. Now, you have MRI also. MRI is magnetic resonance imaging. Over here, you use non-ionizing radiation. So, X-ray which is there, it is ionizing radiation, it is harmful for us, but here in MRI, it is non-ionizing radiation, and the magnetic field that is being used to find the cancer in the living tissue, and everything like that. So basically, three of them are there for internal organs, and they have their specialization as well. CT for three-dimensional image of internal organs, MRI for finding problems in the living tissue, but anyway, three things are there. Okay, so over here you have got the options radiography and MRI. This is correct. MRI and CT, that is also correct. Vidal test, Baba, it's for typhoid. Why can it be here? How can it be here? It is not the one, right? And now MRI and Vidal test. Vidal test again would be the wrong one. So your only options would be A and B, and A and B option is 3. So the answer for this question is going to be 3. Please make sure you know what are the exclusive function of these, these things as well, separately as well. Yeah? Okay? Cool. Ma'am, can you please explain previous question? I can, Prince, can I ask you a favor? If possible, can you wait, since anyway you are a student over here, you have been with me throughout, can you wait until the end? I'll repeat everything together, because I think for some children it's been too late, right? So if possible, yeah? Yes. I hope that works. So guys, give me a thumbs up if at all it's all cool. Let's go ahead. Only 13 more questions are there. Let's solve it and finish it up. Thumbs up needed. Yes, perfect. Thank you. My phone is heating. Please don't watch via phone. It will burst out. Please keep it aside. Okay, watch via YouTube. What do you say? Laptop or things like that. It will be really helpful for now. Don't use your phone if it is heating. Okay. Malignant malaria is caused by, guys, it is a simple question. Malignant malaria is caused by Plasmodium falciparum, Plasmodium rubrum, Plasmodium malariae, and Plasmodium vivax. What do you think is the answer for this? What do you think is the answer for this? Karthik says it is 3, Manoj says it is 1, Anand says it is 1, Bhima says it is D. I am getting A, B, C, D options are. Guys, tell me one answer. Okay. Spurti says it is 1, Rihan says it is 4, Spurti says 1. Yes, Raghavendra, dear, what help do you need? Macha, tell me. So most of you guys are giving me the options as 1, 2, 3, 4, which is obviously not correct way of doing it. Okay, so the answer for this particular question is going to be, guys, malignant form of malaria means the one that is most dangerous. It is going to be what? A. It is going to be A. That is 1. Plasmodium falciparum. You have Plasmodium malaria. You have Plasmodium vivax in your NCRT. You have Plasmodium falciparum. Plasmodium falciparum, the one with the longest name, is the one that has that causes what? Malignant form of malaria. It ends with M also. Okay, so Plasmodium falciparum is the one that causes the malignant form of malaria. Do not get confused. This is one of the easiest question out there. Okay, so the answer for this is 1. Is this clear to all? Ma'am, short notes. Raghavendra, I'll try my best to arrange something for you guys. Yeah, but it will take some time, I think. But yes. Ma'am, we don't see. How can that be possible? No, you have seen it, probably have forgotten, okay? P. falciparum, easy but confusing. So always remember, the one has a bigger name is the one that causes the bigger trouble. Okay, P. falciparum, bigger name. Okay. Yes. Next question that is there, question number 49. The drug prescribed to the patients who have undergone organ transplant is dash and is produced by dash. Come on. If at all a person who have undergone organ transplantation, okay, what drug will the doctor give to that particular person, and what is the organism that produces it as well? That is the question. Lahari says 1 is the answer. Hasina says 1. Prakash says it is C. Quadric, it says 1. Noha Kazi 1. Sanjay also says 1. Prakash says A. And Chetan says 1. Spurti 1. Neha. And Muhammad Javeed also says, Muhammad Javeed has a different answer. Okay. Raghavendra, Dista. Passionate says 3, Anand says 1, Karthik says 1, Varadraj says 1. Most of you guys are saying 1. Smita is hello there, hello, hello, hello. Ravi Chandra, Deepa. Okay, Deepa is also back. So guys, the answer for this particular question is going to be 1. Cyclosporin A and is produced by Trichoderma polysporum. The best way to remember it is just remember it as a story. For example, Jasmine Ma'am's kidney went off. Okay, so now I am going to take it from Norman. Norman will be giving the kidney. Okay, the problem with the kidney is that my body feels it's foreign kidney. My body is going to react against it. My immune cells are going to be super active and is going to go ahead with graft rejection. I want to stop that. I want to make sure my immune system is made to go a little low. Okay, it is suppressed a bit, which is why I'll need what? Immunosuppressants. Immunosuppressant. Cyclosporin A is going to be immunosuppressant, and cyclosporin A is something that I get from Trichoderma polysporum. Trichoderma polysporum is a fungi that is going to produce this, which is an immunosuppressant, which is why the doctor is prescribing me this. I hope, Noman, you're happy with having a kidney there and there itself. Yes, and my children, I hope it's clear to all of you. Kidney class, no sorry, chemistry live class will be there tomorrow morning, I think. Yes. Yeah. Yes. Once more. Okay. So basically, my body will be rejecting anything that is foreign. And I want to stop that. So what do? Immunosuppressants are given. It suppresses my immune system. Okay. Cyclosporin A, which is there, it's an immunosuppressant. It will suppress the immune system and will allow my new kidney to be right there and could help me well as well. This cyclosporin A is taken from a fungi, Trichoderma polysporum. Now guys, I want to ask you one very important question. Everyone who gave me the correct answer, I'll give you a chocolate if at all we meet anytime. Okay, you can just, if at all you see me anywhere in Bangalore, you can just come to me and ask for chocolate. Okay, along with the question number. So anyway, the question goes like this. Which type of immunity is responsible for graft rejection? Which type of immunity is responsible for graft rejection? This is there in Human Health and Diseases chapter. Come on. Any idea? Madan, you are asking me not to take class tomorrow, or you are asking me, will you be taking class tomorrow? Ma'am, is it a fungi, right? Yes, it is a fungus. Okay. Cell-mediated response. Exactly, Rihan. You are getting a chocolate if at all you see me in Bangalore. Okay. So cell-mediated response, that is basically it is being done by the T cells. You know there are two types of immunity: acquired immunity. There are two types of immunity: humoral immunity, mediated by your B lymphocytes are there. They produce antibodies and blah 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 things. Cell-mediated immunity, which is done by T cells, and these T cells are the ones which is responsible for graft rejection. Graft rejection. Okay. So please remember that. It is cell-mediated immunity. Rihan, you are in the chocolate. The second person to get the chocolate will be Shankar, and then Lahiri. And the chocolates are ended. Okay. So will you take or not? So I think, dear, I thought actually I have the live class tomorrow, but I think chemistry ma'am has the live class tomorrow morning. Okay. I'll have to check. If you join the WhatsApp channel, everything is going to be updated there. Okay. 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 Perfect. Next question, question number 50. Guys, only 10 more questions there, Macha, and we're gonna finish everything. I understand some of all questions. Thank you so much. Okay. Read the following statements and select the correct option. Statement number 1. Biocontrol refers to the use of biological methods for controlling plant diseases and pests. Statement number 2. Trichoderma species are effective biocontrol agents for several plant pathogens. What do you think is the answer for this? Statement number 1. Biocontrol refers to the use of biological methods for controlling plant disease and pest. Is it? So usually what do we use? We spray pesticides. But pesticides are chemicals. They can damage our soil and everything. Okay, which is why we should be using biocontrol agents. Sridhar says the answer is 3. Quadric says it is 2. Rihan says it is 2. Lahiri says it's 3. Why a confusion between 2 and 3? Why there's a confusion between 2 and 3? Most of you guys are saying 2 only. Vijay, Kalpana, Anjali, and Komali, and Geeta, Smita, Bhavana, Manjula. Okay, so the answer for this, let me check it out. Biocontrol refers to the use of biological methods for controlling plant diseases. Yes, instead of using pesticides, we are going to use organisms that are going to eat the pest. So we do not need pesticides anymore. Now, statement number 2, Trichoderma species are effective biocontrol agents for several plant pathogens. So you might have seen Trichoderma polysporum is there for cyclosporin A, is there in human beings. How can it act as biocontrol agent in the case of plants? Please remember, in your NCERT it is clearly given, Trichoderma species which are there, they are biocontrol agents for plant pathogens as well. Okay, that could be a different one, but yes, Trichoderma is definitely is a biocontrol agent. And what does it do? It basically controls the fungal pathogens. Okay, there might be some fungal pathogens affecting the roots of the plant. The Trichoderma is going to act like a biocontrol agent over them. Even though it's a fungus, it is going to kill other fungus that causes the trouble. So I'll say both the statements are correct. Statement number 1 as well as statement number 2 are going to be correct, which is why the answer for this question is going to be 2 or B. Okay, so for all of you who have given me the other answer, I hope it's clear to you now. Okay, is it clear? Both statements are true, exactly. So guys, can you give me a thumbs up if at all everything is super clear to you, whatever I said until now? Thumbs up, Macha. Yeah? Yes. No, Mantish, tell me what happened. Okay? Yes. Thank you. Let's go ahead. 51st question. Match the column 1 with column 2 and choose the correct options given below. Streptococcus, Penicillium, Methanogens and Anabaena. What do you think is the answer for this particular question? Any idea? Shankar says the answer for this question is 3. Okay, I'll just move aside. Okay. And Sanjay says it is 3. Bhavna says it is 3. Apurva says it is C. That is 3 basically. Smita says it is 3. Shankar says 3. Geeta. Okay, thank you. Noha, Anjali, Shakshi, Sridhar, Bhima, Vijay, Smita, Ravi, and Lahari, Chidambar. Okay. Yes, so most of you are saying it is going to be 3. 3 is what? This. Let me check it out. Streptococcus. So match the following. What is the rule? First match A and D first. Okay. Streptococcus, Penicillium, Methanogens, Anabaena is given over here. Free living nitrogen bacteria, clot buster, source of antibiotic, biogas production. Okay. Now we're going to solve it. Streptococcus is going to go with clot buster because Streptococcus produces streptokinase, which is going to burst any clots in our blood vessels and will save us from myocardial infarction or heart attack. Okay. Now, Anabaena, which is there, it's a bacteria. Okay, it's a bacteria. Particularly, Baba, it is going to be a cyanobacteria. Yeah, and it is going to be there for nitrogen fixing. Okay, so Anabaena goes with 1. Now, Penicillium and Methanogens are given. Penicillium notatum. Okay, Penicillium is a source of penicillin, and penicillin is what? An antibiotic. Methanogens are what? Methanogens are one responsible for biogas production. In that particular case, what will be your answer? A goes with 2. Okay, just a second. Okay, by the way, when we matched this and this, did we get our answer? A goes with 2. A goes with 2. Okay, and basically D goes with 1, and we have already got our answer. So that is why I say please match A and D first, and then you will get the entire answer. 3, 3, 3, 3. Exactly, it is 3. Anyone has any doubt over here in this particular question? Okay. Streptococcus, what does it produce, guys? Can you tell me the name of the thing that is being produced by Streptococcus? Streptococcus, it produces something that is a clot buster. What is the name of the thing that is a clot buster? Any idea? This was so easy. Yes. Streptokinase. Exactly. Streptococcus produces streptokinase. Okay. Penicillium, which is there. Penicillium notatum, which is there, which is producing penicillin. Which bacteria's growth was it inhibiting? I am asking you another question. Okay. Penicillium notatum, which is there, which was producing penicillin. Which bacteria's growth was it inhibiting? Do you remember the name of the bacteria? Penicillium notatum is basically the mold. What was it inhibiting? Any idea? Staphylococci, exactly. So do not get confused between staphylococci and streptococcus. That is why I asked you this question. Okay, the staphylococci or staphylococcus, that was a bacteria that was being, Alexander Fleming was working on it, and one day he forgot to wash the dish, and the next day he was like, oh my god, the staphylococci might have increased so much. But the next day when he saw that, oh my god, there is a mold being formed, and this mold is not allowing the growth of staphylococci. He was surprised, and that's how he found penicillin. Okay, so please remember staphylococci has so much L, and Alexander Fleming also has L. So that is that, and streptococcus is something that is different. Cool. Ma'am, can you speak Marathi? What all questions are asking? No, I cannot speak Marathi because I do not know it. Okay, so yes. Clear na? Macha, give me thumbs up if it all everything is clear. Everything, if it is clear, just give me thumbs up. Let's go on to the next question. Come on. Yes, perfect. Ma'am, my mother tongue is Malayalam. Yeah. Match the contents of list 1 with list 2. So over here you have list 1, over here there you have list 2. What is the rule of match the following? Read all the options carefully first, then only go ahead. Okay. Bioreactors, downstream processing, recombinant PCR. I don't know recombinant what is there. Okay, anyways, all the options are over here. Insulin produced by rDNA technology, and vessels which converts raw material into specific products, detects mutated genes in protein suspected cancer patients, not patients, and involves separation and purification. What do you think is the answer for this? Come on. Okay, everyone is saying 3, 3, 3. Sakshi says it is 3. Rihan says 3, and Apurva says C is 3. Okay, ma'am, you talk in Kannada. Nanu Kannada kalita idini macha. Okay, and Anjali says 3. Ramya says 3, and Bhavna says 3. Most of you guys are saying 3, 3, 3. Some of you are saying 4 also. Why 4? Listen, bioreactors are going to be what? They are going to be vessels which converts raw material into specific products, right? A goes with 2. Can I find my answer with that? A goes with 2. I can at least eliminate the other options. Eliminate it. That is going to help you out. Okay. Now PCR, polymerase chain reaction. It is there for amplifying the content of the DNA. But I do not see amplification over here, which means that I am going to get confused. Okay. So what is the thing that is, the other thing that I could know? Other thing that I could know is probably downstream processing. Downstream processing means basically separation and purification. So B goes with 4. B goes with 4. I just have one option, and with that I have got my answer. By the way, PCR is not just for amplification. You need to understand that. For example, I have a pathogen in very small amount in my body. Okay, this pathogen, I want to find what is the problem in my body. So basically, I take my DNA, whatever is there, I amplify it. I will be able to amplify even a little portion of the DNA which is there. For that also, I need PCR. So it can be helping me in finding little amount of DNA, or basically sometimes it can also help me with finding mutated genes in the protein suspected cancer patients. So mutations, if at all it is there, basically the PCR will be able to detect it. It will not be having a complementary base pairs and stuff like that, right? So yeah, so downstream processing goes with this, and recombinant DNA is what I assume is over here. So insulin produced by rDNA technology is a recombinant DNA. So the answer for this particular question is going to be 3. Is this clear to all of you? Is this clear to all of you? Guys, IPL, you tell me, in IPL, whoever wins the match, will it benefit you in any ways in KCET examination? If not, please restrict to talking about IPL after the end of the class. You know, I love to have a fun talk with all of you guys, but it is exam time, macha. We are having fun talk, little bit much of fun talks over here. At the end, we will talk about it. Okay, RCB, CSK, whatever is there. Cool? Yes. How many questions are there? 52. 60 questions are there in your paper, so basically 7 more questions. Okay, let's go. The part of the plasmid that codes for proteins involved in the replication of PBR322 plasmid is what? The part of the plasmid that codes for proteins involved in the replication of PBR. Do you know the answer for this? Bhavana says it is 1. Rihan says it is 1. Thank you for being so considerate, Raghavendra. Okay. So Noha Kazi says 1. Neha Venkatesh says 1. And ROP. Rihan says 1. Sakshi, Hemant, and Norman, Rihan, Bhavish. Most of you are saying it is 1. Sahana, Tamanna, Melo. Okay. Samarth, Smita, Spurti, and yes, Deepa, Ravindra. So what is the answer? Most of you are saying 1. 1 is the answer. ROP site is the one that produces the proteins. How many of you need an explanation of this? How many of you need a quick explanation of PBR322? If yes, just say yes. Please explain. Just say explain. I'll just explain. Come on, first. This is important, okay? You might get a question on PBR322. Yes, okay. So I'll just explain it to you. Listen, here you have PBR322 matcha, okay? PB or 322 is right over here. The things that we know about a plasmid is that it will be having an origin of replication, so ORI site goes over here. It will also have a site for production of a protein which is super important for replication, and that is ROP. That is why the question is part of the plasmid that codes for protein. It is ROP. Okay. It will also have what? Ampicillin resistant gene. That means if at all it has this particular gene, it will be resistant to the antibiotic ampicillin. I can grow the plasmid anywhere even if ampicillin is present because it has the resistant gene. Similarly, it will have a tetracycline resistant gene right over here. Tetracycline resistant gene right over here. Done. Now, you need to have 8 cloning sites. That means 8 enzymes which are there, they are going to cut over here. Okay, 8 enzymes which are there, they are going to make cuts over here if at all needed. Let's go with the first enzyme. So 1 pista shake PST1, okay, and 1 vada pav PVU1. PST1 and PVU1. Okay, with that 1 pista shake and 1 vada pav, I went to what? I went to eco class. So this is eco R1 site, and class 1 is the next site that is there. And then I realized actually I entered the Hindi class because the third period was Hindi. So Hind 3. Pista shake, 1 pista shake, 1 vada pav, and I entered to eco class, but then I understood it is Hindi class, third period is Hindi. Okay. And then I bammed into what? Salma Miss. Okay, BAM H1 and Sal1. And I gave her 2 vada pav so that she becomes happy. I gave her 2 vada pav. So PVU2. So always remember PST1, PVU1 sites are over here in the ampicillin resistant gene. Eco R1, Clavin and Hind 3 is over here. BAM H1 and Sal1 is over here, and PVU2 is right over here, which is why this is going to be important. Okay, please remember this is going to be helpful. Okay, cool da. Okay, instead you can say maga. Maga means bacha, right? Choto mone, right? Ma'am, give the break. 53rd question, 7 more questions. You can take eternal break out there. Okay, cool. OMG, the trick I know. Okay, so yes. So shall I go ahead? Please remember all this thing. This is very, very important out there. And when is the next live class, Bhavish? When is the next class live of me? If at all you're asking about me, I think it's day after tomorrow morning. Okay. Now question number 54. Guys, can we go ahead with question number 54? If yes, can you give me a thumbs up? Called Makkale. Okay, looking gorgeous. Thank you, guys. The reason I call macha is like a friendly slang that we call friends. So assume you guys are my friends. That is why I call like that. I love it. I love to call it that way. But yes. Okay, guys, come on. Question number 54. To isolate DNA from fungal cells, bacterial cells, and plant cells, the enzyme required are. What do you think the answer is for this particular question? 1, Bhavana says 1, okay. Quadric also says it is 1, Noha Kazi. Ravindra says it is 1, yeah. Rihan says 1, and Anand says 1. Sakshi says 1, Rihan says 1. Yes, Akhi, I forgot that also. Okay, most of you guys are saying 1, some of you are saying it is 2 also. It is not 2, it is going to be 1. Listen, why 1? Because you want to isolate the DNA from fungal cell. Fungal cell wall is made up of what? Chitin. So you need to break open the chitin with the help of chitinase. Bacterial cell, the cell wall is made up of what? Peptidoglycan. For peptidoglycan breakdown, you need what? Lysozyme. Okay. Then you have plant cell. Plant cell wall is made up of what? Cellulose. For breaking down cellulose, what do you need? You need cellulase. You need what? Cellulase. So basically, your answer for this particular question is going to be 1. Why not the other options? Because over here, cellulase has been mentioned for fungal. No, wrong. You have to look for the order over here. You have to look for the order over here as well, which is why the answer for this particular question is going to be 1. Cool. You are from Kerala, ma'am? Yes, my roots are from Kerala. I was born and brought up elsewhere in north. Okay. So yeah. Is this clear to all of you? Question clear? If yes, just give me a thumbs up. Okay, just give me a thumbs up. We'll move on to the next question. Yes, perfect. Let's go ahead. Question number 55. In mature insulin, which of the peptide is not present? C peptide, A and B peptide, A peptide, or B peptide? What do you think? What do you think is the answer for this particular question? No, I'll definitely do that. Okay, don't worry. Kerala Kuti. Yes, Kerala Kuti. Okay. Yes, passionate. Yes, I remember you telling me that. Okay, most of you guys are saying it is one only. What is one? One is basically C peptide. Is it C peptide? So listen to me. In the case of normal insulin, you have A and you have B peptide, and over here both of them are attached with the what? C peptide. And this is basically not the mature form of insulin. It's not the mature, so I'll call it pro-insulin. Okay, pro-insulin means it is not mature. That means it is not in the working condition. Now, if at all I want to make it in the working condition, I have to cleave, or basically I have to remove the C peptide from here. Now, what will I get? I will get only two strands, A and B, and both of them will be attached with the help of disulfide bonds. What is left behind over here? What is left behind is a C peptide, is going to be a C peptide. Now, I'll call this as what? Mature form of insulin without the C peptide. So, in mature insulin, which of the peptide is not present? The answer would be C peptide, and all of you gave me the answer for this. Yes. Okay, na? Yes, na? If yes, give me a thumbs up, my dear children. We'll move on to the next question. I think this is an easy question, right? Imagine, guys, you are solving KCET 2025 question paper. Yes? Okay. Let's go ahead. Perfect. Question number 56. A scientist wants to produce virus-free plant in tissue culture. Which part of the plant will he use as an explant? Any idea? So, scientists want to produce virus-free plants out there. Virus-free plants in tissue culture. Which part of the plant will he use as an explant? Mature stem, axillary meristem, apical meristem, mesophyll cells, and over here you have the options to choose from. Come on, guys, choose. Okay, now Neha says C is the answer, Rihan says 4 is the answer, Bhavish says 4, Manjula says 3, okay, Anand says 4, and Apurva says C, and Rihan 4, Ravindra 3, Noha Kazi 4. Why there's a confusion between 3 and 4? Why there's a confusion between 3 and 4? Most of you guys are still saying 4. 4 is what? B and C. 4 is B and C. B and C. Yes, 4 is the answer for this question. Listen to me carefully. What happens in tissue culture? We basically take a part of the plant, and now since we have taken the part of the plant, it's now X part, right? So we'll call this X plant, right? X plant we have taken. This thing is put in a medium where it is going to grow, and is continuously going to divide into multiple cells out there. I'll call this whole thing as what? A callus. Now this callus is being taken into the next place where I put it in a hormone which is going to be helping me with the formation of roots. Again, I'll put it in some other hormone which will be helping in the formation of the stem. That is how things are going to go. Okay. Now, from where will I take these plants if at all I want to produce virus-free plants? That is important. I am going to take it from two places. Wherever there are meristematic cells. Meristematic cells means the ones which have ability to divide continuously. There are two places. One is apical meristem, which are there at the tips. Okay, this is shoot apical meristem, other is root apical meristem. I can take it from the shoot apical meristem. Now you also have axillary meristem. So the place from where a leaf is coming, here also you would be having some meristematic cells which can give rise to a branch or a leaf or anything out there. This is known as axillary meristem. So either this or this I'm going to take up for production of virus-free plants. So just imagine if at all this particular plant is affected by virus, still the apical and the axillary meristem will be free of it. Okay, so I'll be using only those, so which is why the answer for this question is going to be 4. Since I am able to see that most of you were able to give this question and some of you were not, I believe now it is clear. For those of you who feel that it was not clear, is it clear now? If it's clear, can everyone give me a quick thumbs up? If it is clear, can you give me a quick thumbs up? Yes, perfect. Now we are not clear? Okay, what happened, Macha? Tell me. Okay, cool. Next question. Some strains of Bacillus thuringiensis produces proteins that kill insects. Which one of the following is not killed by proteins of Bacillus thuringiensis? What do you think is the answer for this? Some strains of Bacillus thuringiensis produce proteins that kills insects. Which one of the following is not killed by proteins of Bacillus thuringiensis? Cotton bollworm, tapeworm, tobacco budworm, armyworm. This is also very easy. Very, very, very easy. So Kalpana says 1 is the answer for this. Peace says it is tapeworm. Okay. Rehan says it is 2. Anand says it is 2. Lehari says it is 1. Okay. Manjula says 2. Sakshi says 2. Navya says 2. Sahana says 2. Bhavna, Lehari. Most of you guys, Norman also says Savitra. Okay. So the answer for this particular question, Vaishnavi is also there. Atif says 2. Yes. The answer for this is actually 2. And those of you, Heyman, Baba, Uday, everyone. Tapeworm is the answer. Why? Because tapeworm is not coming under insects. You need to understand that. Tapeworm is what? A platyhelminthes. It's a flat worm. They do not come under arthropods, whereas cotton bollworm, tobacco budworm, aardvark, they're all insects out there. Okay, and you know, Bacillus thuringiensis, basically they killed insects, right? Different type of insects. So the answer for this particular question is going to be 2. So it's a very simple concept. They have just asked you in different format. Okay, and if at all we get confused, that will be bad, right? So I hope this is clear now. Is it clear now? Is it perfectly clear now? Yeah, na? Give me a thumbs up if it's clear. Yes, perfect. So let's go ahead with the next question. Question number 58. Which one of the following population attributes contributes to increase in the population density? Yes, Macha. Thank you, Macha. Okay. So which one of the following population attributes contributes to increase in the population density? Come on, Macha. This is very, very, very easy. Very, very easy, okay? Natality and emigration, mortality and immigration, natality and immigration, mortality and emigration. Which one increases the population? I'll tell you the understanding of it. Natality means birth. Okay, birth rate. Emigration means if organisms are moving out of a population, that is emigration. E for exit. So if at all they're moving out, obviously that is not going to increase the population size, right? Right, yes. Mortality and emigration. Mortality means death. If people are dying, how can you increase the population, right? So yeah. 3, 3, 3, 3, 3, 3, 3. See, okay. So 3, 3. Most of you guys are saying 3. Some people have get it wrong. So Rihan, Apurva, Vivek, Mr. Raghavendra, and Mr. Raghavendra, please check Macha. And Bhanushri, Ganesh Patil is also there. Hello, dear. Mahesh. So answer is natality and immigration. Natality means birth, and immigration means if at all organisms are coming into the population out there. Right? So the answer is that. Sharan, Shankar, Sahana, and Neha, Hemant, Vivek, everyone. Sakshi, Noha, Prince. Cool? Yes, Matcha. Let's go ahead with the next question. Try. Question number 59. Everyone done with the other question, right? First of all. Done with the other question? If yes, give me a thumbs up first. Okay. Let's go. If 8 individuals in a laboratory population of 80 fruit flies died during a specified time interval, the death rate in the population during that period is what? The death rate in the period is what? Any idea? If 8 individuals are there in a population of 80 fruit flies and we are saying that 8 of them are dying, what will be the death rate in the population during that particular interval? So you have to do 8 by 80. The new organisms which are dying or basically being formed, you can put at the top, and the initial population size you have to put at the bottom. What will be your answer for this? It will be 0.1. 0.1, right? Individual per time interval. This is very important. 0.1 per individual per time interval. This particular thing has to be mentioned. If at all the unit is not correct, then also your answer would be wrong. But what do you call it? Surprisingly, they have not given much things to confuse you apart from this. So please remember that. Here sometimes the unit can be wrong. Look at the unit. The thing can be same. The unit can be different. Yeah? Shreya Gowda, we have already solved the first few questions. Dear, this is a question paper that I am solving. So in the previous live also I was solving it, but due to some technical issues it got stopped. From the 33rd question onwards we have been solving here. Okay? So you can check it out. Okay. Guys, last question for the year 2025 KCET question paper. Come on, solve. Choose the correct sequence of steps involved in decomposition. Fragmentation, mineralization, humidification, leaching and catabolism. Fragmentation, leaching, catabolism, mineralization, humidification. Fragmentation, catabolism, leaching, humidification, mineralization, and the same goes. What do you think is the answer for this particular question? Need update, I'll check, Amalu, okay? Okay, so last question, the answer, Anand says it is 4, Sakshi says it is 3, is it? Okay, Anand says it is, wait, Sharan, RIP, okay, just. Neha says it's true, Quadricate says true, Pragya says it is 4, and Chetan says it's 2, everyone. Most of you are giving me the correct answer, but some confusion is still up there. Fragmentation, leaching, and then you have catabolism, FLC, and then you have humidification and mineralization, FLCHM, FLCHM. Okay, so fragmentation, leaching, catabolism, mineralization and humidification would be a wrong thing. Fragmentation, mineralization would be a wrong thing. Fragmentation, catabolism, leaching would be a wrong thing. Fragmentation, leaching, catabolism, humidification, mineralization would be the correct thing. So the only answer for this question is going to be 4. So those of you who said who answer is 3 and all, I hope it's clear now. Is it clear? This concept is important. Do you know the concept? Do you need an explanation of this concept? If needed, I can help you with that. Thank you for taking the class, Macha. You're most welcome, Macha. Okay. Bhima, please put your mobile away and don't watch from there. Okay. Guys, do you want an explanation? If you want an explanation, say explain. I'll explain. Yes, Asma, correct. So everyone who gave me the correct answer, I'm super proud of you. Everyone who gave me the wrong answer, I hope you have learned. Yeah? Yes, perfect. Thank you, Hina and everyone. Okay, explanation needed. Fine. So those of you want to go, you can go, dear children. Do not waste of any bit of your time. And those of you want to stay for any doubts, or as I said, I'm going to explain this fragmentation, leaching and everything. If you want to stay, you can just stay back. Okay. So thank you so much for everyone who have joined and who are gonna go. Wishing you the best. Lots and lots of love. Probably we'll come meet again very, very soon with a FLT. This was a previous year paper. I am going to come up with a full-length mock test paper. It's a new questions. Be there for that live. The timing will be shared with you in the WhatsApp channel. The PDF of this will also be there in the WhatsApp channel. Okay. Thank you so much, Mr. Raghavendra. Melo, love you too, dear. Thank you so much. Bhima, bye-bye, 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 bye-bye. Okay. Thank you, Amalu. Okay, everyone. Rihan, thank you, thank you, thank you. Navya. And Passionate, love you too, dear. Vijay, thank you, everyone. Chalo. So let's solve the fragmentation, leaching and everything one. First one is fragmentation. Listen, the only target that we have from here is that we want to break down things. And the first process for that would be fragmentation. Why fragmentation? In fragmentation, what are we going to do? We are going to break down the big things into smaller fragments. So it's like a physical breakdown that you're having. It's more of not a chemical breakdown, but a physical breakdown. And for this, you will require what? You will require some organisms which you call as detritivores. Detritivores as in some organisms that can feed on dead and decaying matter, but necessarily they are not decomposers. They can be earthworms. They can be what? Earthworm. So earthworm, basically they take in food and they poop it out. While they poop it out, obviously they're going to break things down, right? So this is going to be known as fragmentation. And while they poop it out, you also need to understand that there might be some things which can be water soluble, and they can be inorganic as well. They can be inorganic and water soluble. Listen, the complex things are always organic. The simple things are always what? Inorganic in nature. That means organic things break down to inorganic things. So here, with the first step, we are not saying that all the organic things convert into inorganic. We are saying that some of it might be there. Some inorganic things could be there. They could be water soluble also. These inorganic water soluble things can go deep into the soil and leach there. So basically this process is known as leaching. This is known as leaching, where they go deep into the soil and penetrate out there. Please remember two things, water soluble and they are inorganic in nature. Cool. Now the next thing that we have is next thing that we have is we need to break this thing down out there. Okay, so we are assuming that we are going to start from the starting point onwards. We'll break down the organic things into what? Completely inorganic things. For that, the process that is being used is known as catabolism. It is known as catabolism. So fragmentation done, leaching done, and finally you're going to do catabolism. Over here, you are going to need another organism which is out there, not detritivores. This time you are going to need what? Decomposers. And the decomposers are Facebook, that means fungi as well as bacteria. And all of it, remember, they are acting on the detritus. Detritus means dead and decaying matter, okay, until this particular process. Now, when you talk about catabolism, we just assume it's breakdown of organic to inorganic, but you need to understand that I can divide it into steps. For example, when I'm converting this, I will reach a halfway place where I am going to get something that is known as humus. I am going to get something that is known as humus. It is dark, amorphous substance which is being formed in the soil. And please understand, it's like a midway. From organic to inorganic, you get something in the middle, that is humus. And this process is known as humification. This process is known as humification. Here, no one is acting on the detritus. The decomposers are doing their job, and in between you are getting it. So this process is the process where you are not acting on any detritus. Please remember it. Okay, this is given in your NCERT. It's confusing also. There is no direct action on the detritus. Okay. Now, always remember the structure of it is amorphous, and basically it is dark. It is not crystalline in nature. It is not crystalline in nature. You can get questions on the basis of that. Finally, the humus is going to break down. Finally, the humus which is there, it is going to break down into what? Into inorganic things. And that process is known as mineralization. And that is going to be a very slow process. Very slow process. And finally, we say catabolism is completely done. So in your NCERT, we feel that catabolism, humification, mineralization is something different. But actually, in actual sense, while you remember, you do not need to remember it like that. Just imagine when catabolism is happening, midway you say humus is being formed, that is known as humification. And then when the humus finally breaks down to inorganic things, you call this as mineralization. It's a very slow process. It's a very slow process. That's all. So fragmentation comes first, where you're physically breaking down big things into small things with the help of detritivores. Now, whatever is soluble and what is inorganic, okay, it will leach into the soil, that is known as leaching. Whatever is left behind is being acted upon by decomposers, and the decomposers do the rest of the job. Then you have humification, formation of humus, and mineralization. Done. Love you for teaching, good mother. Thank you so much, Raghavendra. PDF notes you will get in the WhatsApp channel. So children who stay back over here and okay. One of the good, Ma'am, good explanation. Thank you so much, dear. Okay, Comet ke liye bhi kuch karo. So Comet ke liye we are doing the Comet ke launch pad and stuff like that. But yes, you have so many series out there. Please check it out. Okay, everyone. So guys, I hope it's clear. And if at all you're worried about Comet K, one very important thing. There is Comet K30 crash course which is ongoing. The classes have started on 6th of April. If at all you want a full coverage, make sure you go ahead and take it up. It's for 2499, and if you feel that crash course is not needed now, I just need to solve questions, then you have test series. 12 full-length mock tests are there, just available at 999 rupees. Cool. So please check that out as well. Cool. Thank you so much, Geeta. Guys, if you have any doubts, you can ask me in the paper that you had done, solved now. If you, PS, I remember you were there with some doubt. If you have the doubt still persisting, you can ask me right now. Okay? Ma'am, you teach Comet K class. In Comet K, there is no biology, da macha. Okay? Thank you, Asma. Love you too. Okay? And thank you, Geeta. Okay. Ma'am, when is your next class? Ma'am, when is your next class? Navya, Karthik. So I think next class will be there day after tomorrow is what I believe, morning 9:30 AM. So mornings are for female teachers. Yes. So, yeah. Yeah. So tomorrow or day after tomorrow, I might have a class. Tomorrow, I think it's chemistry in the morning. Please join the WhatsApp channel. There you will get all the updation regarding the classes and everything. It will be really good if at all you could join it. Yeah. So, no free series for Comet K, no dear, not as of now. Okay, we had lowered down the price before, but yeah. Free series as in YouTube series, yes, it is. So 15 days, things are ongoing, but post that, definitely you will be having more of Comet K things as well. Okay. Hari Prasad, I don't have any idea for cracking KCET, what to do right now. please target the high weightage chapters if you can and solve at least their PYQs. It's going to be super important. Yeah, chapter wise high weightage chapter list is being given to you. Check it out, it will be really good. Okay, so day after, I think from, I have to check, I think it's soon gonna start. Dear, I have to check the, what do you say, the revision classes. Okay, I'll let you, I will update you with that as well. So children, I think that's all. And yes, okay, so I'll just end the class wishing you the best. Guys, if you have any doubts, put it down in the comment section, post the live, and my back is paining, so I'm going. Okay, please take care of yourself. And ABHYAS KCET test series is also there. If you want to take it up, make sure you take it up really fast. Okay, it's available for 999 rupees, and do not miss out on it, and you'll get the everything in the WhatsApp channel. Check it out in the notes and everything. Bye, lots of love.